003. Okay. Yep, we're going to go ahead and get started then. Thanks everybody for making it here. We'll get right into it. Today we're going to have kind of the two sides of the coin of mycobacteria testing here at the state of Nebraska. First, we're going to have Gene Philby, who is a MT, not an MLS. I'll make note of that. It's my mistake. Gene is pretty instrumental in the TB testing that happens here for the state. I'm going to let her kind of describe her process for TB testing. And then we've got, of course, Christian Bertrand to kind of talk about the state side of TB surveillance. So I'll get this PowerPoint up here for Gene. Okay, I think everybody should be able to see that. And so, without further ado, Jean, I do want to make sure that everybody can hear Jean when she's talking over there, though. Can
everybody hear me? Hopefully.
Yes.
Yes.
Okay, great, great.
All right, Jean, the floor is yours. You want me to go?
Yeah, go ahead. So first of all, I want to thank everybody. The state TB program entails many different people. We couldn't do it without everyone. So I want to thank the Douglas County or the health departments, the state TB program, all the laboratories throughout Nebraska. So it does take all of us to help our patients. So first of the thing, I'm going to go through some specimens. The obvious sputum samples we're going to get primarily to rule in or rule out M. tuberculosis. Since Nebraska Medicine does function as the state TB lab for Mycobacterium tuberculosis, once a patient has been identified as a positive tuberculosis, we then become the health lab for that, and we are sent all of the follow-up cultures from any of the health departments in the state. And so that is what we're going to function at, kind of dive into that a little bit too. So of course, our main one is sputums. We also have other respiratory cultures, BALs, bronchwashes, gastric lavages, urines, bloods, bone marrow, body fluids and tissues and abscess. One of the things that we always ask is to make sure that we get actual specimen. Swabs are not recommended, and we usually will reject those due to some of the unique qualities of mycobacteria. Swabs are not good or sufficient material for culture. Stool samples, kind of for obvious reasons, it has an enormous amount of bacterial overload, so please do not ever submit those. And so this kind of gives, if you want to, look at this, this is kind of the quantities and what we usually recommend for those. Now, blood and bone marrows, we're going to be using the Bactec Mycolytic F bottles. It's a blood bottle. Bone marrows, we add a certain amount of bone marrow directly to that. Typically, we put some fresh in. You can also use an EDTA. So transport, of course, getting it to the lab as soon as possible. You must keep it refrigerated. Since we do function as the public health lab for the state of Nebraska, many times we get follow-up cultures. So it's the follow-up cultures, it's always best to send one, make sure that it is actually positive for the smear is actually positive. If it is, there's no need to send the second and third times 3. Just treat and then start over again. So it is important to comply with the shipping regulations for safety. If you are one of the clients that will send an actual positive culture, that needs to be sent as a category A because we do not know whether it's TB or not. This provides safety for everybody that's involved. Okay, so the first thing that we're going to do once we receive the specimen into the lab, we typically will do the processing. It is required, we have a cutoff time of about 8 o'clock in the morning. So it needs to get into the microbiology lab by that time. Otherwise, it will be pushed off to the next working day. So mycobacteria have some unique qualities. Because of the high lipid content called mycolic lipid layer in their cell wall, they are very difficult to stain. They're very slow growing. So we typically will incubate these up to 6 weeks and maybe longer. The treatment time is longer. And again, going back to the swabs, mycobacteria, because of their lipids, will stick to those swabs. They're very hydrophobic. And so that is a little bit difficulty in our isolation of that. So mycobacteria also require proteins for their carbon sources. They're obligate aerobes, meaning they need to grow in the presence of oxygen, but they are enhanced by the addition of CO2 environment. Again, we process samples and set up Monday through Friday. If needed, Saturday or Sunday, it must be stat, and it has to be approved by our medical director or associate director. We go through a decontamination and digestion process, and this is typically done on all respiratory cultures and any non-respiratory cultures that may have a gram stain that has bacteria or organisms, and or if there's growth in these cultures. So it's kind of nice to know if you're sending us samples, whether they are growing bacterial, but we'll take care of that when we get it. So, and we also have validated urine cultures for the use of our midget tubes. And I'm going to dive into the midget. So, and every culture does have an initial smear that goes with it. And that is usually reported out same day once we get it. So just briefly going over the digest and decontamination. So it goes on simultaneously. The digestive part is the N-acetyl-L-cysteine, which breaks down the hydrogen sulfide bonds in the mucus, so it liquefies it. Then the decontamination part is happening with the sodium hydroxide. So this helps by killing or reducing non-mycobacterial organisms. We also have a sodium citrate. It functions a couple of different things. It helps to bind heavy metals. It also helps in reducing specific gravity because of the lipid content. It helps bring down the organisms into a pellet easier. Another type of decontamination is oxalic acid. We do this after the sodium hydroxide portion. This helps in patients having a high Pseudomonas overload in their cultures, such as cystic fibrosis patients. And this process decreases the incidence of Pseudomonas. The next step is centrifugation. We use a refrigerated centrifuge. One of the things that's very important when you are processing is to have, for safety reasons, is to use centrifuge cups with rubber gaskets. Since we don't know if TB is there, when you are spinning that down, you have a pellet, you're concentrating, and it's always good to use safe laboratory practices. So centrifugation is going to concentrate the organisms, and sometimes we will centrifuge if we have large amounts of body fluids just to concentrate those. And like I said earlier, staining, we have two different types of stain. One is a fluorescent stain, which is an Ormino. This one is sensitive, but not specific. So anytime we have a positive fluorescent stain, we have to counter stain with the Zeal-Nielsen. That is our definitive stain, our acid-fast definitive stain. It's specific, but unfortunately, it's not as sensitive. So some of the types of recovery media that we use, Again, we go by regulations from the CDC. We use both a liquid and a solid media for recovery. I think a lot of people use the Midget 960 system. Sometimes there are smaller versions. This has a 7H9 broth. One of the things that mycobacteria need is protein for their carbon sources. So all of this media that we use has a higher content of proteins. Now the midget has, like I said, has a 7H9 broth. It also has a fluorescent indicator at the bottom of the tube. The media has infused oxygen, and since mycobacteria obligate aerobes, they will utilize that oxygen. And when the oxygen level goes down, the fluorescence goes up. So that's what the machine is going to detect. We also add a supplement, a Panta. That is a combination of antibiotics and antifungals. We also have a supplement which contains oleic acid and albumin. Of course, albumin is a protein source. Our solid media that we routinely use is Lowenstein-Jensen, and that's an egg-based media. It is somewhat selective. It has malachite green in it, which for some reason inhibits the growth of bacteria. Usually, we will also use other types of, such as a 7H11 agar. Now, the chocolate agar is always added to certain specimen types, and I'll kind of get into that when I talk on the next slide, but it does help to recover Mycobacterium haemophilum, and rapidly growing bacteria will also grow on chocolate. One of the things that as laboratorians, you will recover rapidly growing mycobacteria from routine bacterial cultures. And typically they will grow within 3 to 4 days on blood or chocolate plates. So kind of always keep that back in your mind. When you're doing gram stains, they'll be very wispy, beaded rods. Another thing that I always try to have people, when I teach them, is when you actually put the organism onto a saline, when you're doing your gram stain, it'll float because of the high lipid content. So kind of always keep that back in your mind if you're ever run across that. So incubation time. So initial cultures are typically incubated for up to 6 weeks. We used to do 8 weeks, but we did some validations, and I think this, the APHL or the CDC now recommends 6 weeks. So we're going to incubate it with the midget tubes. The midget tubes are incubated at 35 to 37 degrees, and it monitors it every 60 minutes. In this particular case, when the indicator is positive, we would typically right away do a Ziehl-Neelsen. So that's our definitive stain. So many times we have mixed cultures, we have pure cultures, we might have just bacteria only. So we'll do different things for these different scenarios. So if it's just bacteria, we'll re-decontaminate it again. If it's mixed if it has acid-fast bacilli plus bacteria, we will sub to selective media and then re-decontaminate it again. But if it's a nice positive, just acid-fast bacilli seen, we'll subculture to 3 Lowenstein-Jensens, 7H11 agar, and a chocolate slant. One of the Lowenstein-Jensen and the chocolate agar goes into 25 degrees Celsius because that will help recover some of the slower growing or and also rapid growers that have to have that temperature. Now, the Lowenstein-Jensen is our routine solid media of choice. They're incubated again for up to 6 weeks. We read those once per week. Also, in initial setup, we set up a second LJ and a chocolate slant And these are added specifically for skin, soft tissue, lymph nodes, joints, bone marrow, bone specimens, and which typically on the outer skin regions and joints, more of the rapidly growing mycobacteria grow better, and that's why we incubate some of those then at the 25 to 33. So more of a room temperature type. So once the slants are positive, we would need to do a Ziehl-Neelsen for growth to ensure that it is truly a positive. And we again then would subculture these for more growth to 2 more Lowenstein-Jensens and 7H or 3 Lowenstein-Jensens and 7H11 and 1 chocolate slant. And the 7H11, for those of you who do acid-fast bacilli cultures, 7H11 agar is very good for more of a rapidly detect Mycobacterium avium intracellular complex, or MAC. They grow very well on 7H11 agar. So some types of identification that we have. So initially from specimen. Now the Gene Expert TB rifampin assay, or an amplified TB probe by Cepheid, is FDA approved only for sputum samples. I know that ARUP and some others have validated for other specimen sources. We are currently working on validating the use for bronchial washings and BAL. So that will be kind of a lengthy process. So once we actually have a pure positive acid-fast bacilli, we have a couple of ways that we can identify them. So our first one that we can use directly from the midget tube. Also, we can do some from a pure culture from the slants. This is our real-time PCR. This is a lab-developed test, and it was developed by our associate director, Dr. Yulan Chan. She has done an absolutely amazing job in getting this underway for us. It does identify M. tuberculosis complex and M. avium complex. This was developed to replace the AccuProbes from Hologic that they discontinued. So we are just so grateful that we have this test. It's very excellent. It's an excellent test and very specific and sensitive. We perform this test weekly on confirmed positives, either, like I said, from the midget or from the slants. Again, we only do this once per week. The reagents are costly and the test is very time consuming. Our second method, and we can do this on midgets, slants, typically we primarily do them on known positive pure cultures. This is our microalign identification. And it's a sequencing. We use specific assays, primers, for these certain regions. This is, again, a lab-developed test. We typically use the RPOB primers, but we also can do 16 ITS primers. Again, we perform this weekly. Again, it is a lengthy process because we have to kill the organisms. Then we have our molecular virology, our molecular micro take care of the sequence, the the primers and the PCR side, and then we send the product over to our molecular diagnostics lab where they have a sequencer there. And then molecular diagnostics sends it back to the AFB lab for reading out of the sequences. So it is a kind of a lengthy process. And again, we always use our basic methods to correlate these other tests by growth rate, pigmentation production, and colony morphology. They need to be consistent with what we have identified. Susceptibility testing, and I think a lot of people who do mycobacteriology testing Some will do their own susceptibility testing. Many of us send most of the mycobacteria species other than the TB complex to ARUP. We send all of our Mycobacterium tuberculosis complex to California Health Department. This is under the auspices of the CDC, so they do a lot of work for the CDC. Then they will also send to Michigan for genotyping, and that's also required for epidemiology purposes. One of the nice things, if you want to use this method to being sent to the California Department of Health, we are in a consortium with Nebraska Public Health Lab. They have the DOT license for category A to send out these known TB cases. This is for safety reasons and just the safety of everyone. So if there are any additional susceptibilities that are required, not performed at ARUP, we can send to National Jewish, and there's University of Texas at Tyler. There's sometimes, numerous times, especially with rapidly growing mycobacteria such as M. abscessus, that ARUP is unable to do certain antimycobacterial agents. So lastly, just a little safety stuff. We utilize a biosafety 2 to biosafety level 3 practices. Our room is negative air pressure. The rooms are monitored with alarms. We use a biosafety hood that has negative air pressure. And again, it's equipped with alarms. It has HEPA filters. It has its own dedicated air handlers. And everything, once it goes through, everything gets vented out on the roof. So it has its own dedicated air handlers. We always wear N95 masks when we are processing specimens, when we are working with positives. We also wear a front covered water resistant gown, of course, gloves. Some people wear masks when we process samples and cultures. Now, again, getting back to the centrifuge, it's double gasket. There's a rubber gasket on the lid, and each centrifuge cup has its own rubber gasket. And of course, then you'll have your specimen cup. So this is going to ensure the safety, since we do get a lot of Mycobacterium tuberculosis. This is something that's very important to keep not only our laboratorians safe, but our community safe as well. That is pretty well it for my quick, but again, we work together with the county health departments, the public health departments, Nebraska Health, and also with the TB program. And a lot of communication. So thanks. If there's any questions, I'm always happy to help.
Thank you, Jean. Like I said, Jean is a wealth of information when it comes to TB testing. Are there any quick questions?
Hey, Jean, this is Pete in the Public Health Lab. Thanks for your presentation. I have a question about NAT testing, nucleic acid amplification testing. As it is recommended for the first respiratory specimen, for instance, on a patient. Do you automatically reflex to a NAT test if you don't get an order from a physician, or do you wait for a physician's order?
Thanks, Pete, for that question. I was hoping somebody would kind of talk about that a little bit or ask that question. So whenever we get a sputum sample in, so Pete is talking about the amplified TB probe. That's what we use initially for identification from a sputum sample. The physician can order automatically if they suspect, meaning if they have chest x-ray symptoms, they can automatically order an amplified TB probe. Since it's only FDA approved for sputum samples, those are the ones that we will accept. And again, if the medical director okays it, we will do others with disclaimers. So yes, they can automatically order it. The other thing is that if a sputum sample is positive for acid-fast bacilli, we will reflex that
okay.
order automatically.
Okay,
So
so
thank you for that question.
So if a physician forgets to order the NAT test on a sputum that they suspect is TB patient, you still have to wait for their order? I mean, can you automatically order it yourself, or do you still have to call them and ask them to put an order in?
So the question was that if the physician doesn't order an amplified TB probe or a NAT test, we will not automatically do it.
Okay, that's my question. Okay.
But if the smear is positive,
right,
we will reflex that order.
Yeah, I get that. Okay.
Okay.
Very good. Thank you, Jean.
Thank you, Pete.
Any other questions for Jean before we open the floor to Kristen? Okay, Kristen, you take it away.
Okay, thank you for having me. I'm Kristen. I'm the nurse coordinator for DHHS TB program, and thank you, Jean, for a great presentation, and thank you to our partners at Nebraska Public Health Lab. As Jean said, we work tandem together and are in very close contact to make sure we're providing appropriate lab testing for our presumed or active TB patients. Just a couple of loose ends. I do not have a presentation today. We do need approval from us if we are going to be going out and collecting sputum. I know that doesn't affect you guys directly in the lab, but just know that if we have someone that we are going to test, we just need approval at the state level. If we do have someone that does come back that is rifampin resistant off the gene expert, that means we have a presumed multi-drug resistant TB case, which is a very big deal to have. It's a very rare thing. I want to say it's 2 to 3% in the US and requires very expensive treatments, bedaquiline, which is over 30 grand for treatments. So, and it's a public health threat because we have people where we don't have first line TB drugs available to treat. So if that would come up, please notify us immediately so we can go ahead and act upon that. If you would hear about that in any shape, form, or way. And when we do get our lab reports back from NPHL, I do send them out to our local health departments, and the local health department will report out to the physician that is caring for the TB patient. Whole genome sequencing, Jean mentioned this briefly. We are doing that on all our with all of our drug susceptibility testing, as there's been problems with some of the test kits with pyrazinamide. So if you are seeing a pyrazinamide lab result coming back on, you probably won't see it on drug susceptibility testing, but we will get you a whole genome sequencing report to help identify any mutations, especially in that PNCA gene, which is which shows the PZA likelihood of being susceptible to pyrazinamide. And if you are using a private lab such as ARUP, we still can go ahead and do the whole genome sequencing or work with our lab to make sure that can be done, as that is a clinician decision how that will be handled. We didn't talk too much about our MDDR testing or rapid drug susceptibility testing. If we have someone that we suspect is, going to be drug resistant, maybe an exposure, or we have suspicion that they're a contact to someone with drug resistant TB. So that is an option, and that is a clinician order, or we can suggest it from the state level to the clinician to make sure it happens. The genotyping is quite helpful for contact tracing, to link cases, to help our contact investigation. So you may get a call from me at your lab if you do have a culture growing, and we do need to chip back to NPHL so they can send it out to Michigan. So we've done very well. We were sitting at 100% last year, which is just unheard of. So I want to appreciate all the labs across the state for your cooperation and great job ensuring to make that happen, to make sure that happens. And other than that, I just want to say thank you to our lab partners across the state. We cannot do this work without you. And that's all I have been.
Thanks, Kristen. Are there any questions?
Kristen, this is Pete. Could you share with the group how often you've seen rifampin resistance in Nebraska?
Like I said, it's pretty much very rare. So it's going to be probably
Okay.
we don't have numbers we can report on because we don't meet the 6 or greater criteria per year. We have seen rifampin resistance here. It is a rare finding, and it is a big deal if we do have a situation as such. It is a real
Sure.
threat. So just be on guard. Like we have to treat every specimen per what Jean said with isolation, proper isolation guidance, but it is an ongoing issue not only locally but nationally as well.
Yeah, it seems to me we might get one a year at most. I mean, it's not very common that we see it from what I could gather, thinking back.
Yeah, it's a rare finding,
Yeah,
and we see various forms of, resistance. I'd prefer to have isoniazid resistance versus rifampin because that's
yeah,
easier to deal with, but we don't get to pick and choose. We have to be ready to deal with whatever comes at us. I'm
sure.
very appreciative that we have Gene Expert available to give us a heads up, so faster results, better treatment, and getting people taken care of because we want to get them on a proper regimen right away as we have such limited options with TB medicine in this day and age.
Right, right. Okay, thanks, Kristen.
Yeah, Kristen, thanks for that. I just wanted to add, so like you said, the amplified TB probe, the gene expert, that's why it's very important for physicians to be aware that that is there. It will detect that rifampin. And I think the numbers that we've had so far in our lab, I think at least one, if not two, detected by the gene expert, the rifampin resistance.
And just to throw out there, Nebraska and TB, we did have a 20-year high reported out last year via the HAN. We're getting very close to publishing our 2024 annual report. It was not significantly higher than what we've seen, but TB numbers have been rising nationally. So just something to keep your eye out. We know TB is a threat. There's an outbreak in Kansas City, which is nearby. So we know the threat is real. So always we want to do everything we can to do every resource we have to, make sure we're getting people tested timely, treated appropriately, and doing thorough contact investigations to make sure we're treating our patients and keeping the risk to a public very minimal. So
Kristen, how many cases this year? We're down from what we were last year?
I don't know if I can report out, but I think we're going to be a tad lower than what we were
Tad lower?
last
Okay.
year. A tad
And it-
lower, but we still have plenty of activity. It has not slowed down significantly. So just keep that in mind that it is something that we will see across the state. Even in our rural parts of the state, we see TB. TB does happen.
Yeah.
did we have any cases that came up from Kansas that you know of or no?
Per the Han, no. That's a nice thing
Okay,
about the TB genotyping is we can identify links not only
right,
locally but nationally. So in that Han, it did state we don't have any links to the Kansas City outbreak.
good.
Kristen, I don't know if you saw Justin Frederick's comment here for Douglas County mentioning an
Yeah.
increase.
Yep, I believe it. It has been very busy across Nebraska, and Douglas County is where we do have most of our TB cases, and it is unfortunately something we've seen, not only in Douglas County but across the U.S. where we've seen increases, and it just requires more manpower, more resources, to deal with this. And for those of you that are in TB prevention and control efforts, it takes a lot of time and many of our patients have many social barriers to, just, getting by every day, doing very simple things such as food, water, electricity, let alone medical care, getting medicine, knowing how to navigate the U.S. healthcare system. So, yes, I do recognize that it is an increase. And we see that every day. And like I said too, it does show this nationally as well. That's what we've been dealing with as well. So
Okay, thanks a lot. Thank you, Jean and Kristen, for talking to us about the TV program. That was very interesting.
Thank you.
Thanks, guys.
So real quick, moving on, I just want to mention that measles testing is now being done in-house at NPHL. I think I saw Emily on the call. Emily, I don't know if you want to elaborate on testing. We're no longer sending PCR to Minnesota. Serology is still being done at reference labs.
Yeah, so I can expand on that further. Not only are we and have been fully live with in-house testing at NPHL for a while now, we are no longer sending out to Minnesota. That order code is MEASLE, which I can put in the chat right now. It's orderable in New Alert. If you are needing measles testing, please still let your local health department know. So we are supporting the state's public health funded measles testing, but we're also working with a partnership with Regional Pathology Services to offer this on a patient pay fee for service opportunity as well. So if you have patients within your facility that are not meeting the requirements for state funded measles testing, you can seek that testing through Regional Pathology Services. I believe a lab alert went out on that last week. And that testing will still be performed here in state with a rapid turnaround time. Appropriate specimens for that are going to be either nasopharyngeal or throat swabs in viral transport media or universal transport media with a paired urine. If anyone has questions on that, please let me know.
I will have the most up-to-date collection and shipping guidance available later today. So if there's no questions on measles, we'll just move on. We'll do our roundtable real quick here. Getting kind of close to the hour. So we'll start with HAI. I think Brianna.
Yes, I'm here.
Okay.
Yep,
Hi.
Dr. Oshoff wasn't able to be on. He has another meeting. Just passing this along for him. CRE and C. aureus outbreaks are still ongoing, and just as a reminder, if there's any suspected C. aureus that's identified at your lab, just make sure those get sent over to NPHL. That's actually all I had. I'm passing it along, but if you have any other HAI related questions, I'm happy to bring those back to the team and get those answered for you.
Great, thanks, Brianna. Respiratory illness updates.
Yeah, hey Ben, this is Derek. So I'll keep it fairly brief as I don't have a whole lot to update on. Haven't seen any increases in flu, RSV, and SARS-CoV-2, at least to this point. So not too shockingly about flu and RSV, but haven't quite seen an increase in SARS-CoV-2. So at least to date, haven't seen that quote unquote summer wave that we've seen in the past. Not to say that we won't, but just wanted to point that out. Also, I'll put this link in the chat, but almost a month ago to the day, we sent out our annual summertime respiratory disease haunt. So I just wanted to put a plug in for that to this audience. It goes, covers a little bit about MERS, since in the summer we want to keep that on our radar, and as well as our summertime influenza specimen submission recommendations. So again, I'll throw that link here in the chat, and then if anyone has questions, feel free to let me know. Thanks.
Thanks, Derek.
I can just piggyback, give a wastewater update here.
Great.
So we do test for respiratory pathogens using wastewater surveillance for SARS-CoV-2, flu A, flu B, and RSV, and also subtype of for flu A, H1, H3, and H5. Right now, as Derek said, we are seeing something similar trends. What we see clinically, like everything is quite low, no unusual spikes or anything seen through the wastewater surveillance data as well. NPHL is currently also working on expanding to other targets like norovirus and AVD68 in the next couple of months. And also we are looking to onboard measles. It's still too early. NPHL has to validate the assays, but that is something we want to do through wastewater surveillance as well for measles. That's all I have. Thank you.
Hey, thanks a lot. Any questions regarding respiratory illness or wastewater testing for those targets? Okay, we'll go on to enteric outbreaks. I am not sure who's doing enteric outbreaks today. Do we have?
I will be doing
Sandra.
it today. So the multi-state backyard poultry outbreak for Salmonella is still ongoing. We currently have about 3 cases. The multi-state outbreak from eggs has officially closed as of last week. We don't have any local outbreaks going on at the moment, but I do want to mention that we are starting to see cyclospora cases. So just a reminder to send cyclospora positive specimens over to NPHL. And that's all the updates I have today.
All right, thank you. We'll just keep moving we got Jeff up next, arbovirus testing.
Jeff is out, so I'll be giving update on behalf of Jeff. So we have 13 cases of tularemia to date and 1 West Nile virus case. And there have been 6 positive mosquito samples 6 pools of positive mosquito samples for West Nile virus. And tick activity is still going on and still on above average level. And we have 18 alpha-gal syndrome cases so far, and we are still investigating about 6 or 7 of those. So we might see more cases moving forward. That's all I have for arbovirus.
Thanks, Samir. Did you say 18?
Yes.
Is that higher than normal?
Yeah, we are seeing a lot more. Well, we don't have the past number of cases, so this is the first time we are doing it. So we will see how that goes.
Curious. Thank you for the update. Kristen, back to you. I don't know if you had anything to add.
I do not have any additional updates.
Okay, thanks a lot.
Thank you.
We'll do the STD program next. We have representative. No news
Dennis
is good news.
Lashinsky is out today. So I know Brady Pham is on the call, but I don't know if he had any updates. I don't have anything for HIV or viral hepatitis today.
Okay. Okay, I don't know. Nothing for STD. Okay, and then we'll finish off with the lead testing.
No updates from lead today.
Updates for lead today. Okay, well, just a couple of house cleaning items here. Becky and we're getting the gram stain challenge ready, the challenge set ready, and you might have seen the notice that the live workshop registration had opened. There are limited seats, but we'll be doing 1 in Omaha and 1 in North Platte. So kind of all the big training projects that we do coming up. And then at the very end here, I'd just like to mention that Emily McCutcheon has been promoted to the deputy director of the NPHL, and Amanda Barling has been promoted to lab manager. So moving and shaking at NPHL. I don't know if there are any other new staff or open mic if anyone wants to address anything before we close out.
Ben, I will just real quick say that the Nebraska Public Health Laboratory is currently has 2 job openings. If anyone knows of someone looking for a position, we are looking both for a molecular technologist as well as a chemistry technologist within our group. The job can be found on the UNMC job posting site. So, thank you all.
Come work with us. Okay, so that's it for July 14th, 2025. Next meeting will be August 11th, same time, same place. Thank you, everybody, for joining.